Yeah, thank you for the introduction. Ka sachaljenju, ja ni gavaju по-русски. Я очень рад быть в вашем прекрасном городе и рассказать вам о моей работе. Okay. So you understand I have to practice much more. Okay, so Taras convinced me to compose a series of 10 lectures from Monday to Friday, and I hope by Friday you would still be here. And I'm trying to, as I just was trying to say in Russian, which you probably did not understand, I'm trying to make it interesting for you. And Taras kindly referred to our work as out of the mainstream. And we like to work out of the mainstream because there is no competition. We work 7 days a week, 24 hours a day, but outside of the mainstream, and that is a lot of fun. And if I can get my things to work now. So not to plunder again, I just put it on the slide. I simply copied this from the web page, so don't you think I was able to do this myself. So that is the topic of my series. And before I begin, I would like to present to you my group, a group of very dedicated young and not so young people working together on these various topics. And this is the locations of one of our retreats in the Swiss Alps. And I noticed not only is St. Petersburg a very beautiful city, this is also very beautiful, and you probably do not know, but that is only 20 kilometers away from our institute. I don't know why this is on the... Staufen, yeah. I don't know why this is on a poster of a Russian company, but I'm very pleased. So I really feel at home. Thanks very much. So acknowledging these people, now in passing I will mention them when I report about our own work, but I will also, of course, have to mention many other colleagues that did more interesting and more important things than we did. And I'm trying to build a sort of story for you to understand why it is important to consider problems of immunity in the general sense, and in particular problems of adaptive immunity, that is how the immune system changes the state of a system in order to cope better with a second or a third challenge when genetic or bodily integrity have been attacked by outside invaders. But you will probably not be surprised to hear that this function of protecting genetic integrity is not very modern. It already originated about 2 or 3 billion years ago with the first unicellular organisms, and I will give you a couple of examples in the first lecture. I will try to make a couple of definitions, and I'm taking the risk of being a bit boring in the beginning, but I'm trying to throw in one or the other sentence so that you will be interested in coming again tomorrow and until Friday. I will be discussing, for example, why sex is good for you. And sex is good for all of us, and if you stay until Thursday, I will give you interesting instructions for your parties at the weekend. Okay? And I prepared some lectures that will fill about 9 hours, and I've left 1 hour free for you to ask me questions, and I would urge you to interrupt me even now if you think I'm unintelligible, even in English. So I'm trying to do my best, but you might have different levels of knowledge, and I'm quite happy to answer your questions. And I would like you to go away and hopefully say to yourself, okay, I've learned something. And in order to do this, if you don't understand my reasoning, just interrupt and ask. And I'm using this wonderful electronic device, but I'm also quite used to using the blackboard. So if there is anything that I need to explain in more detail, as I said, interrupt me. Now, the first couple of slides will be very trivial, and you might say, why did I come here and rather not stay outside in the park in that beautiful weather? But nonetheless, I think I need to start with some basics just to get you the idea, and then we can work from there, and it will be more and more detailed. I will even show you some nice videos of some experiments that we did that I like very much, and I hope you will also too. So what is immunity all about? When you think about this in general terms, it basically means preservation of organismal integrity. And this may sound rather trivial, but that of course for any living organism that is a very important matter. It has to preserve genetic identity, so not to be invaded by bacteriophages if you are a bacterium, or by retroviruses if you are a multicellular organism that somehow destroys your genetic makeup or even interferes in your physiological function. Or you might want to protect yourself in terms of protecting your bodily integrity if some multicellular parasites come, some worms or whatever you might be infected with. So this is the area that immunity covers, immunobiology and immunology proper. So it's a very fascinating area of work. You can work at it if you are a medic, you can work at it if you are an evolutionary biologist or molecular biologist. There are plenty of questions still unanswered, and I will address some of them towards the end of the series. Now, in philosophical terms, immunity is all very simple. It's about distinguishing self from non-self. If you know who you are, you can decide that something else is not you. So the Greeks put it very simply. You probably know the Oracle of Delphi, where this was one of the sentences that was put on top of the big wall when you enter this temple. So know thyself. If you don't know yourself, then of course you are in trouble. And this is the English version of this Greek inscription that I found, of course, on the internet, but it's copied from a high-rise building in a German in a German town nearby Freiburg. So how does it work? So when you think of yourself as engineers, biological engineers, and you have to fulfill the task of distinguishing self from non-self, you have basically three strategies. And you will find, or you perhaps know already, that all three strategies are actually used in nature. So you have one possibility, and the first possibility is you have to if you focus on recognizing self, all you don't recognize is not self. And when you think about this, is this a useful strategy? We tend to think that the immune system focuses on the recognition of non-self. So all self is basically ignored, and everything that is not self is considered to be potentially dangerous because it is non-self. Now, when you think about building a system as an engineer and how to train the system, it's not like a neural network, it's a biological system, and so you have to think about how you train your system to distinguish self from non-self. Then it's not so trivial. Because if you focus on a mode that recognizes self, you have to make sure that you recognize all self. So no single structure in the organism must be left untouched in order to instruct your system to recognize it as self. Now think of a developing embryo or an organism. You all know that we have in the order of 20 to 30,000 genes that make plenty of proteins, sometimes in many different forms from one gene. So we have plenty of proteins, plenty of proteinaceous and non-proteinaceous structures that are being made by the biosynthetic pathways in our body. And we all know, of course, that we develop. So when we start as a fertilized zygote, we have a different program of gene expression than when we look at maybe a muscle in the heart or an epithelial cell in the intestine. So they all draw from this basic genetic information, but they're all morphologically and phenomenologically different. Now, because the phenotype is different, that means the composite of cell structures also must be different. Now, when you begin your life as an embryo and you develop into a stage where the immune system first starts to operate, then you want to make sure that the immune system learns about your self composition, but it might well be that some structures were only important in the early embryonic period, and once you're past this, the immune system has no chance of seeing this again. So some people, for example, interpret the appearance and maintenance of certain tumors to this problem, that early in embryogenesis, some structures are being made, but then they go away, and some embryonic type tumors in later stage of life then use these structures, and the immune system cannot recognize it because it wasn't trained to recognize it as self. Then if you focus on non-self, this is an equally important problem, because you cannot imagine a system whereby you can make ways of recognizing every possible non-self structure. Because it can be a carbohydrate, it can be a structure from some algae, from some bacteria, or from some parasite, multicellular parasite, Plasmodium, for example. So that is also very difficult to envisage how this might work, and we will address some of these problems during our series. And there is a third way of doing it, and that is in fact the way that our immune system uses. It combines the two strategies. It is trained, or it's built to recognize not only self, but also non-self. And now, considering what I just said, this must be a horrendously complex problem to solve for the immune system, to ensure that the immune system protects genetic and bodily integrity without harming our own body, yet be able to and ready to attack anything that comes from the outside. So this is a very interesting problem, and immunology therefore has evolved into some sort of general topic where many different disciplines can find home. Comparative zoology, for example, when you compare immune systems of different animals from invertebrates to vertebrates, and we will discuss some of these interesting issues there. Molecular biology in terms of how proteins interact, how proteins are diversified from a limited amount of genetic information, and we will address some of these problems during the course of the lectures, and you will see that you can find plenty of things to do should you ever be interested in continuing this subject later on. Now, I was trained as a medic, and I was a pediatrician before I decided I wanted to do basic science. My clinical training was mostly in hematology and oncology, and I learned the hard way that it was very difficult to treat young patients with these aggressive tumors, be them leukemia or some embryonic type tumors. And I decided I wanted to solve that problem from scratch, and I was somehow drifting into immunology. I still haven't come anywhere near solving this problem, but I stuck with this system, and I'm trying to convince you that it has many aspects. Here is one. When you are a clinical microbiologist and you have to type a particular type of bacteria, and I've taken this from this particular paper by Gibbs and colleagues, and you will find whenever I have a reference here, it's not our own work. And I was trying to be good to everybody. Should I have made a mistake, I apologize already, because I hear it's all being taped, it will be put on internet. And I should also say that I've been taken to court for making reference to data of other people without proper acknowledgement. So that was a bit scary when Tavas wrote to me, it will be taped, and I thought, oh God, this is a lot of work. Actually, it was a lot of work, but I was trying to give credit, of course, to all the people that did all these fancy experiments that I'm trying to use to build my arguments. So this is now, you can read the top, this is now a bacterium that has this wonderful name, Proteus mirabilis, because it's a very interesting type of bacteria, and it can swarm on plates. Some of you might have seen this. And the way to type different isolates of these bacteria is to put them next to each other and observe how they behave. If they belong to the same serotype, they will mix. You can see that here. If they don't belong to the same serotype, they will stay apart. They will not mix. So even bacteria are capable of recognizing and distinguishing self from non-self, which I find quite extraordinary. And that is now a phenomenon that is being used to type these isolates, so you can distinguish whether one patient is infected with one particular type or another type, and if there is an outbreak of infections, you can very easily see and trace it back to a single source by doing these kind of analyses. So that is a way how bacteria distinguish between themselves. And why would they do this? And this is another topic that I will address. And I was mentioning this already. This will be important for your weekend parties. Immunology has a great impact on ecology, because ecology is all about cooperation. Okay, so you all sit here, and when you go for the exam, you cannot collaborate. Okay, you're deliberately being separated, so you will not be able to see what your neighbor is writing down on his papers. But in reality, of course, you cooperate. Okay, you go to school together, you go to lunch together, you help yourself when studying a difficult problem, and that is, of course, ecology, and immunity has a lot to say about ecology. And this is one of the reasons why these bacteria don't like to mix when they don't belong together, because, of course, they are selfish. And the immune system also translates in some level of selfishness, because the immune system wants to protect genetic identity, and that is only possible by distinguishing from other parasites, genetic parasites, for example. If you think of retroviruses, we want to fend this off. So this is a ubiquitous phenomenon. Just to whet your appetite, I showed you this. Now there is another distinction when you think about this, again philosophical. Some people have distinguished immune systems into two different types. Epimetheus and Prometheus were two brothers that you know from Greek mythology, and one of which is labeled the afterthinker, the other one is labeled the forethinker. So I hope you all belong to this group of people, because you are living a complicated life as students, so you have to think well ahead when you prepare your exams, for example. So, but Epimetheus, of course, you know all the Pandora's box and all that story. So Epimetheus was a man who simply acted and then thought about the outcome. And we like to see ourselves in the opposite way. We think before we do things. It's not always true, I admit, but I think we tend to believe that we belong into this camp. And you can think of the immune system in the same way. The immune system can deal with problems either ahead of time, and some people call the Promethean immune system the anticipatory way of looking at immunological problems, or you can operate by the Epimethean mode. You just wait and see, and then decide what to do. And both of these have their advantages. Now think again in ecological terms, and we are all about, it's all about, another aspect is saving energy. Now if you invest in any way possible in your organism into preparing yourself for an insult that might never come, that is of course a waste of energy. So, for example, I never bothered to learn Russian because I didn't think I would ever come to Russia. Now, you see, I'm paying the price. I should rather have prepared myself, and my wife urged me to say these sentences at least. She wrote that down for me, so I could at least give the impression as if I was able to speak Russian. So this was the Epimethean type, okay? I just waited to see what happens. Now Taras forced me to come, and I wasn't prepared. But now I know, okay? If these lectures go well, he might invite me again, and just in case, I will probably learn a bit of Russian. Now, Prometheus, this is the anticipatory mode. That, of course, invests a lot of energy for things that might never happen. And you all know, and you are used to think of this, because we are, for example, we are making antibodies that can distinguish. Now, if you imagine yourself as a chemist, that can distinguish between stereoisomers of the same molecule. You can't get any better than that in chemistry. And we can make antibodies, or our bodies can make antibodies that can distinguish these enantiomers, which I think is an incredible level of discriminatory capability. But why would you want to do this? Why would you want to be able to make antibodies against these strange molecules? Remember the lunch diner experiments when he was proving this point, that he could distinguish sugars, for example, that had one or the other type. So there's a lot of energy in here, but it wouldn't exist on Earth if there wasn't an advantage. And the advantage, of course, is once you have prepared yourself for a certain type of insult, you can react very rapidly, possibly, and you can fend off severe damage. Whereas with the Epimethean mode, you have to wait and see and then react accordingly, and that might be too late then. Okay, so this is another way of looking at things. And I'll show you something here, which is now all the journals that you might be able to read are full of this particular type of genetic system that was discovered maybe 10 years ago or so in bacteria. And it can be interpreted in many ways. In the papers that you read, the immunological aspect of it have all but disappeared. It's only about biotechnology now, because one can use this very specific recognition system to engineer precise mutations into either cells or organisms or whatever. And it works like this. It was discovered in certain bacteria, and this is a very sort of simplified version of things, but it tells you or gives you a very good example of Epimethean type of immunity. So here's a bacterium that has a particular genetic locus by this complicated name, CRISPR, and it transcribes this DNA that is here in red and in blue into sequence-specific copies, of course, of RNA. So this DNA is copied into RNA. This, of course, you all know. And these RNAs are called guide RNAs, because if foreign DNA is being introduced into the bacterial cell, these RNAs will recognize the same sequence and will guide a particular type of endonuclease, here depicted in green, to form a complex, and this endonuclease will then cut DNA and destroy it. Now, from this sequence of events, you immediately see that this is not Promethean, because it remembers genetic insult that might have happened. So it might be passed on vertically, but it can also react in an adaptive fashion. So imagine a bacteriophage. This is my depiction of a bacteriophage, injecting its DNA into the bacterium, and we'll come to another defense mode in a moment. So this DNA will then integrate into the genome. It will form a lysogen, as you all know from your microbiological lectures. And this is now foreign DNA. And this foreign DNA, of course, might be bad for the cell, because it might lead to production of bacteriophage and lysis of the cell, which, of course, is not what the bacterium wants. So when this DNA comes, the bacterium that survives this attack, and it has other means of surviving, and I'll come to this in a moment, it integrates a small piece of this foreign DNA into its CRISPR locus, and next time round the phage comes, the phage is destroyed by the CRISPR system, because this bacterium, having survived the first infection, remembers the genetic DNA when the second infection happens, and that phage has no chance of integrating and replicating. Now you might say, okay, well, this is very well, but how does the bacterium deal with the first infection? Because if it's killed first time round, there's no point in creating that system. So the system survived because the bacterium has other means of dealing with foreign DNA. And that is an interesting system, again, exploited to the fullest in biotechnology. It makes use of enzymes that either cleave DNA or modify DNA and recognize specific DNA sequences, and you all know this is the restriction endonuclease or modification systems. That has been discovered many, many years ago and has been used, of course, and was the basis for the biotechnological revolution. Now here is another interesting aspect that concerns immunologists throughout their work, which is the problem of statistics. Now imagine our friend, the bacteriophage, now again injects DNA into the bacterium. Now the bacterial DNA, if it carries a restriction modification system, this RM system, is fully protected against the endonucleolytic attack of these restriction endonucleases, because all the restriction sites are modified. So the bases are modified, in this case by methylation, by putting a methyl group onto them. So this is this M here. And when this site, this sequence is modified in this way by the modifying enzyme, the restriction endonuclease cannot work. It's blocked from action. But the bacteriophage's DNA, of course, is naked. And because it's naked and not modified, it will be cleaved and destroyed. And some of these pieces of DNA will then be used in the CRISPR system to create epimethylin immunity. But you can see the problem, of course. If this was a foolproof system, the bacteriophage would no longer exist. Of course, it's an arms race. If the modification system, being a little bit stupid, is fast and modifies the bacteriophage's DNA, then of course the bacteriophage is safe and the bacterium is doomed to failure. Okay? So there is a competition, and that competition prevails through all immunological concepts. They are rarely spelled out specifically, but it's always the case. If you are infected at the right toe of your foot and your antigen-specific T cell that you also have happens to travel in my left arm, there must be ways of informing that T cell of the specific infection in my right toe. And you can imagine now this is quite a long way to travel for a small cell. So the immune system has to solve a number of very important problems, and one of these solutions is to build specialized organs. And we'll come to this later in the presentations. Specialized organs that we refer to as lymphoid organs. Some of them are called primary lymphoid organs, others are called secondary lymphoid organs. And there's an interesting evolutionary trajectory dealing with all these problems. And imagine now a blue whale. A blue whale is a very big animal. It probably doesn't even fit into this lecture hall here. And it has the same problem. It has many lymphocytes, and lymphocytes are antigen specific, and it's being infected, say, in one of its fins, and then the lymphocyte has to travel about 150 meters to get there. This is next to impossible when you think about it. So that means you have to provide a certain number of cells with a given specificity, so to be able to basically cover the big volume, you have to find ways of informing these cells that something has happened, and you have to somehow bring the cells to the site of action. So all these things we will discuss later. So this is why I was trying to summarize this here. That is the first take-home lesson. The first take-home lesson is it's basically an arms race. There is never a winner. If the host manages to defeat an infection, the parasite will find ways to get around this, because it also mutates. So we have here generalized variation in the population, we have genetic recombination, hypermutation. Any possible genetic modification you can think of is being used by parasites, and any possible genetic modification you can think of is used by the host. And of course they just go round and round and round, and sometimes the host invents some extra measure like dedicated immune cells. I mentioned lymphocytes, for example. We'll come to this later. It orchestrates the response in certain ways that we refer to as immune response, but it can never win. But when you think about it, this is often put in evolutionary terms into the Red Queen hypothesis. You might have read this famous novel by Lewis Carroll. So the Red Queen, when she explains to Alice how the system goes. It says, okay, I have to do all the running just to stay in place. That means an organism can only survive if it constantly adapts, modifies its responses, modifies its ways of dealing with the outside world, otherwise it will be extinguished very quickly. And this arms race we will have plenty of things to discuss about. Now, I promised to talk about sex, because I found everybody's interested in sex. There's only two things that make the world go round: money and sex. I can't talk about money, so I have to talk about sex, I'm afraid. So is the emergence of sex very difficult to explain? I don't think it is very difficult. And there are now more and more experiments that point us in a certain direction that make it very clear when you think about it on paper why sex is required. If you don't have sex and you reproduce, your genetic composition, save for the occasional error in copying your DNA, will basically stay the same. So if you imagine your arms race, and if you are genetically static, the winner of the day is the pathogen, because the pathogen decides not to be static. It changes its DNA, it changes its ways of attacking you, and it won't be long before you are gone. However, this is not a very good thing for the pathogen. Imagine a pathogen infecting an immunocompromised host. The pathogen is used to attacking someone that has a functioning immune system, because all its reproductive strategies depend on opposition. So if, for example, a parasite infects a host and it needs, just for the sake of argument, a week to reproduce, and it infects myself, I have no functioning immune system, and I'm dead in 3 hours, bad luck for the parasite, because I'm not a good host. Okay? So the parasite has a vested interest in a good immune system in the host. It may seem a bit paradoxical we come to this, but when you think about it, this is why this arms race can go on. If one of the two keels over, arms race is finished. And we can be certain, because we are complex multicellular organisms that have a very long reproductive lifespan, and the pathogens are normally more short-lived than we are, it will be the pathogen that will win if one of the two decides to change strategy. So here is why we need sex. Sex allows us to recombine genetic information. Because when we think about ourselves in an ever-changing environment, we always have to be prepared for the occasional new parasite emerging on the scene, either through mutation or through importation from some else. Think of HIV. HIV is a very instructive example. HIV was confined to some part in Central Africa, and through ways that we don't have to discuss, managed to escape from there and to spread across the world. Now, the Western world was not used to this particular parasite, so the immune system of a typical Westerner was not quite prepared for this. For HIV, initially that was very complex, because HIV had a difficult time to take hold in the population, because it had to establish some sort of mutual correspondence in deciding, okay, I don't harm you too much, you don't harm me too much, and then we just live a happy life together. But if there is something that you have to be prepared for, it's best to mix genetic composition from time to time, not to go to the extreme, to make it very incompatible, just to be a bit of in overlap and similarity, but add a bit of new things to it from time to time. So that extra bit will give you the opportunity to survive. Imagine this fantastic story that the North Americans tell you when you speak to them, how they defeated the indigenous Indians. They thought they had fantastic strategy, superior guns and weapons, and had ways of managing a large countryside. But the truth of the matter is they simply introduced some viruses that the Indians had never seen before. So the population size of Indians crashed by 95%. But the important lesson for the immune system is that 5% survived. Measles, for example. 5% of the Indian population, for some reason, survived the infection by measles virus. And they could so because their immune system, and we're not going into the detail in this first lecture, because the immune system was prepared ahead of time for some unforeseen attack. And these 5% survived and then rebuilt the population. And that happened all over again. It happens with HIV. You know that there are some genotypes that confer resistance to HIV infection. It has happened when mycobacteria infect a population. It happens with Yersinia pestis. There are all sorts of, or even vaccinia. So there are so many occasions where a parasite or some pathogen comes, wipes out most of the population, but there's almost always a small minority that survives, and from this genetic bottleneck, the population can grow up again. And this is a very instructive experiment published a couple of years ago, once again by Prockhurst and colleagues. So they used little worms, and these little worms are particularly useful for this particular experiment because they can reproduce asexually or sexually. They can switch. They can decide what to do. So these worms don't invest in finding a proper mate when conditions don't change. I think that's a very sensible strategy. So they are hermaphrodites, so they can self-fertilize. So they're genetically basically very simple, uniform. But when there is a problem in the environment, they can switch to sexual reproduction mode, and that means they find a mate that is genetically distinct. So do we. Okay, we are not self-fertilizing, and that will of course be very important later on. Taras mentioned this in his introduction. This is a very interesting connection to the immune system. So what people have done here is they've simply infected these worms with a particular parasite and simply observed what happens. And what they found was that there is co-evolution. Co-evolution means that the infection with a new parasite changes the rate of sexual reproduction. You can see that here. So when evolution happens, you select for a resistant genotype, sexual reproduction goes up until you've reached a particular genetic fitness, as it were, and then it goes down again because it doesn't make any sense, because the environment doesn't change anymore. But when you then infect every time again, when there is a new generation with this particular bacterium, there is co-evolution going on, and the entire population, because it's reinfected every time, increases the rate of sexual reproduction. So it's an energy question. It doesn't make any sense to invest in all this business of finding the right person. No courtship behavior, because you know the environment is not going to change. You can co-evolve, and that is the interesting question that the immune system needs to answer. So the immune system, or the immune system as such, has a heavy influence on evolutionary trajectories that might seem completely remote from the functioning of the immune system. And I gave you this thought experiment before. What happens if a parasite infects an immunodeficient host? That is absolutely terrible for the parasite. So the parasite has an interest in having hosts that have a certain level of resistance. Otherwise, this co-evolution can never work. So now the immune system, this is my sort of interim summary. Is there a clock somewhere? It's quarter past. So I have 5 minutes before we make a break. And I try to learn what the proper Russian word for intermission is. And I only have it from sports, time, but I don't think that's the right word, is it? Half time. Okay, well, never mind. So the immune system impacts our life at various levels when you think about it. And if I made myself clear enough, it affects the individual fitness. That seems pretty obvious. It affects the reproduction rate of the population, because if a population is genetically homogeneous, it might be very vulnerable to a certain attack. It has an impact on the level of the species, because the species might change the mode of reproduction. I just gave you the example of the worm going from asexual to sexual reproduction, a very fundamental change indeed. And then it also has an impact on the ecosystem. It's basically interspecific competition. And it's not necessarily interspecific competition the way we see it. So different animals that are alike, for example, particular types or species of deer, or a bear or a deer, but it can also be a very tiny parasite and a very big animal that falls under this interspecific competition. So some more definitions. You heard about innate and adaptive immunity. This is what you normally read in the textbooks or on Wikipedia. Innate immunity is something that is installed during development and is ready to go when there is a need. It does not arise, this is why this is perhaps called innate, from any previous infection. It does not use any knowledge. But when you think about it, now you're well prepared, I think, and you're a clever group of people. When you think about it, this is definitely a misnomer, because innate immunity is shaped through evolution. My parents passed on genes that allowed survival in their particular environment, and by definition, the genes that make up my innate immune system are shaped through evolution. And of course, not only over one generation, but over many generations. By contrast, adaptive immunity is often referred to as something that prepares the body for future challenge. I refer to this as anticipation. We make, for example, antibodies just in case. Or we refer to it as a Promethean. We're thinking ahead and preparing for a potential challenge. But as you perhaps already guessed from what I was saying, these definitions are very imprecise, and they basically prevent you from understanding the complexity of the immune system. So I'm not sure how, when you sit your exams, you probably have to, in one way or another, respond to these questions. But when you pass the exam, forget about them. And try to look at the system in a more complex fashion, because you will find that these definitions do not help much when you understand the function of immune systems. There is co-evolution, evolutionary trajectories, and it's basically a mix-up. And I'll give you this here. None can live without the other. This is my way of building cartoons. So we have innate and adaptive functions. They normally cooperate. So when one is missing, the other one is also incapacitated. And if the other is missing, this one is also incapacitated. So there is functional interaction. And that simply means that these definitions dividing the two are probably not the most interesting and helpful ones. Now, which organisms will we discuss? That was my introductory hour, and we will have a short break, I understand, now in between before we go on to the second one. I just hope you come back. So which organisms will we discuss? We will discuss flies, mollusks. We will probably not discuss too much sea urchins and things. We will discuss Amphioxus, the lancelet, a very basal chordate. We will discuss lamprey, jawless vertebrates, the sister group of jawed vertebrates like us. And there are various things we will be discussing about jawed vertebrates. Our own contribution, as Taras mentioned, is mainly to the right of this scheme, from jawless vertebrates to humans, because we are interested in the immune system of vertebrates. But we found, because again we were ill-informed when we started all this, and we are learning as we go, we now find that we have to go back from time to time to the other organisms that I just mentioned in order to understand where certain functionalities in our so-called modern immune systems come from. So I will allude to them in the next hour, just to set the stage for the more in-depth discussions that we will have a little later. And before we have the break, two types of antigen receptors are required for the immune system. Of course, you've wondered why I never mentioned how self and non-self are distinguished. Of course, it all comes down to molecules. And these molecules are called receptors. And there are various ways of doing it, and you will very quickly understand why that is a particular problem. We have so-called germline encoded receptors. Germline encoded refers to the fact that they are not changed, they are passed on to me from my parents, and if there is no copying mistake, they will be the same as the ones that my parents had, and I will pass them on to my children and perhaps also to my grandchildren. So this is an inherited line, this is horizontal gene transfer as it were, and there is no change in composition. This will underlie Darwinian evolution. If there is a mutation in one of these genes that does not work well, this will be weeded out in the population. If they all work well, or if there is a beneficial mutation from time to time, it will be selected for. But the important point is we can predict what the genetic capacity is that is required to encode these receptors. Think of your genome. To maintain a genome is very costly. You need many cookies to maintain your bodily integrity. And every gene you add to your complement means more energy. Okay? When I give lectures in Germany, I'm not sure whether you're used to the same things, I always refer to the Mars bar problem. Okay, you can eat so many these chocolate bars to maintain yourself. You know, you need 1800 or 2000 calories per day to keep you going. And how many Mars bars that is equivalent to, and how many Mars bars you need to maintain your immune system. It's about half a Mars bar. Just think of it. So it must be very important for your body. So adding genes is costly. So this is why there was another invention that is called somatic diversification. I think that is very, very, very clever. And we will discuss that in a bit more detail now. Somatic diversification means make many out of one. Use a piece of genetic information and find ways of recombining it, reshuffling it, reusing it, modifying it to make a lot of genetic information out of very little coding capacity. And that, of course, is a very clever trick. It's just a bit like Lego. You have a couple of building blocks and you put them all together in different ways and you have many different outcomes. The problem here is, how do I guarantee quality control? Okay, imagine I've seen many Mercedes and Audi cars on the streets of St. Petersburg. There must be factories somewhere building these cars, I would imagine. So when these cars are built, there is some quality control at the end of the construction line before they're being sold to the customer. Now, Audi probably would not be so profitable and so successful if they would sell shoddy cars. So they probably have a very good quality control before the cars leave the factory. Well, at least that's the idea, I would imagine. Now, the immune system has the same problem, because once the immune system sends out faulty components into the periphery, think of the blue whale, it cannot call back this faulty lymphocyte. If that lymphocyte has escaped from quality control, this is bad news for the entire organism. So quality control is a particular problem when you start fiddling around in the soma, not in the germline. In the germline you can do Darwinian selection, in the soma you cannot. But you have to find ways of solving that problem.